Hi everybody, welcome to Dr. Manny's YouTube Learn Shops. In this presentation we're going to review, as an overview, cellular biology, how cells and tissues grow, cellular body fluids, which can be intra, extra and interstitial, and cellular transport mechanisms. So when it comes to cells, body tissues grow only by increasing the number of cells. And there are lots of cells in many tissues of the body, and they all divide and grow very quickly from our pre-adulthood to the time that we become adults. Now, skin cells or blood cells are dividing all the time, because we're constantly using these cells up. When cells become damaged or they die, the body has to make new cells to replace them. And this process is called cellular division. One cell doubles by doubling into two, two become four, four become eight, eight becomes 16, 16 become 32, and so on. Now, when it comes to cells, there are two major types of cells: the eukaryotes and the prokaryotes. Eukaryotes are typically larger and have a more extensive intracellular anatomy and physiology in comparison to prokaryotes. Now, inside the intracellular compartment of a eukaryote are things called organelles, which is pretty straightforward, I guess, because within our system, within our body, we have organs. Within a cell, the eukaryote, we have organelles. In prokaryotes, the difference is, in comparison to eukaryotes, is that there are no organelles, and their nuclear material isn't encased in a nuclear membrane. Prokaryotes, or prokaryotic cells, are characterized by having no nucleus. So when we look at certain components within the cell, I'm just going to review some selected components that are more common. The first one is the nucleus. Now, the nucleus is an organelle only found in eukaryotic cells. It's fully enclosed in a nuclear membrane or covering, and it contains a body called the nucleolus. And the nucleus contains all our genetic material, DNA, chromosomes, so that we can now pass that on to our genetic offspring. The nucleolus, as I said, is located in the nucleus of the eukaryote. And the nucleolus takes up about 25% of the volume of the nucleus. And its main function is to produce ribosomes, which are essential elements for making proteins in the cell. The cell membrane is a covering. I guess it's almost like skin in us. But the cell membrane, also known as the plasma membrane, is a thin, semi-permeable membrane that surrounds the cytoplasm of a cell. And its function is to protect everything inside the cell so that the organelles are protected and certain things like the cytoplasm remain within the cell. However, the cell membrane, plasma membrane, selectively allows certain substances into the cell while keeping substances out of the cell that it does not want to enter. Now, the plasma or cell membrane, every cell has one, eukaryote. And this membrane has got gates openings, pores, channels, and pumps. And these membranes can allow, depending on the cell, various molecules or substances to enter and leave. And it's because of this selective transport systems that they can move molecules in and out of the intracellular, extracellular, and interstitial space. So therefore, the cell membrane is responsible for controlling the movement of substances from one compartment to another compartment. Intracellular, interstitial space, extracellular. Extracellular, interstitial space, intracellular. Then we have the cytoplasm. The cytoplasm, I guess, could be compared to having your plasma in the blood. Now, the cytoplasm is a thick solution that fills each cell and is enclosed by the cell membrane. And it's composed primarily of water, salts, and proteins. And organelles in the eukaryotic cells are situated in the cytoplasm. So they have contact with water, salts, proteins, and any other substances that they require. Now, the mitochondria, the powerhouse of the cell, is an organelle in a eukaryotic cell, and its function is to perform cellular respiration. And in this process of cellular respiration, mitochondria make energy-producing substances, and the main one being ATP, adenosine triphosphate. And therefore, this means that the mitochondria take up nutrients from the cell, break it down, and turn it into energy. Lysosomes are like our digestive system. And lysosomes are organelles that can be compared to digestive enzymes that break down excess or worn out cell parts. If, for example, a cell is damaged beyond repair, lysosomes can help it to self-destruct and be eliminated. And this process is called apoptosis. And apoptosis is really programmed cell death. Peroxisomes are another organelle located in a eukaryotic cell. And these are membrane-bound organelles, and their function is to break down lipids or neutralize free radicals as a result of oxidation. And again, for example, hydrogen peroxide can be broken down into safer molecules, because if left to accumulate, can damage the cell. When we look at cells, there are lots of different types of cells in the body: muscle, fat, bone, blood, nervous, epithelial, immune, reproductive. And cell differentiation is basically the process by which dividing cells change their functional type or their phenotype. And all cells, theoretically, come from a stem cell, which is at the top of the hierarchy in cellular production. And as they mature, they develop these functions. So cellular composition is often modelled as a hierarchical scheme, with stem cells at the top of the hierarchy, with other cells being produced. When we look at the functions of cells, cells have got different functions depending on what body part or what tissue they're related to. So, for example, muscle cells are related to movement. Neurons or nerve cells are related to conductivity. Together, nerve cells and muscle cells can generate movement. In relation to metabolic absorption, all cells have to take in and use nutrients and other substances from their surroundings in order to function correctly, homeostasis. Secretion can be things related to mucus glands or cells that can generate new substances from substances that they absorb. Excretion, all cells have to excrete waste products. And lysosomes, as we indicated previously, are within all cells, eukaryote cells, and they break down large molecules into smaller molecules and again produce waste products. Then you've got respiration or cellular metabolism, and this is where cells absorb oxygen, aerobic metabolism or respiration, which is used to transform nutrients into energy. And the form of energy that we typically spoke about previously was ATP, adenosine triphosphate. But cellular metabolism, as indicated when we looked at the organelles, occurs in the mitochondria. Then you've got reproduction. Tissue growth has to occur, so cells have to provide more cells. So tissue growth occurs when cells enlarge and reproduce themselves. However, not all cells are capable of continuous mitosis division. Neurons don't regenerate. Myocytes don't regenerate. Certain cells don't regenerate. Communication is important because, again, communication is vital for cells to survive. This is why we refer to homeostasis. Conductivity is basically, as we said, a response to a nerve cell and a stimulus. And typically, stimulation is depolarization. Regenerating or repolarizing is going back to its pre-state before depolarization occurred. Cellular respiration is a set of metabolic reactions in cells of organisms, with the ultimate objective being to convert chemical energy from nutrients into ATP. Now, the energy-using process of metabolism is called anabolism, and the energy-releasing process is known as catabolism. And it's important because metabolism provides the cell with energy it needs to provide and produce cellular structures. Cellular metabolism then is all the chemical tasks to maintain cellular functions. The energy-using process of metabolism is called anabolism. The energy-releasing process is called catabolism. And ATP has got a universal role as a fuel inside living cells. And the objective is for a cell to function, it must be able to extract and use the chemical energy in the organic molecules, such as glucose. Now, when 1 mole of glucose is metabolically broken down in the presence of oxygen, this is called aerobic metabolism. And with aerobic metabolism, 1 mole of glucose produces 38 molecules of ATP, which allows the production of, or the availability of, 686 kilocalories of chemical energy being released. If there is no oxygen, anaerobic, only 2 molecules of ATP are produced for every 1 mole of glucose. So aerobic metabolism or aerobic respiration produces a very efficient amount of ATP molecules, those being 68, when compared to anaerobic metabolism, only 2. Now, there are 3 phases to cellular respiration. Phase 1 is digestion, phase 2 glycolysis and oxidation, and phase 3 the citric acid cycle, also known as the Krebs cycle, also known as the tricarbolic acid cycle. In phase 1, extracellular digestion of large macromolecules are broken down into simple substances which produce ATP. In phase 2, intracellular breakdown of subunits of acetyl-CoA accompanied by the production of a limited amount of ATP and NADH. In phase 3, the production of NADH yields ATP and waste products such as water, carbon dioxide, ammonia and urea. The essential thing is the ATP is produced. Communication is essential for everyone, including cells. And in order to maintain a stable internal environment, or homeostasis, cells need to communicate. And they do this by using hundreds of kinds of signal molecules. Insulin is an example. Chemical communication can be primary, and in primary chemical communication, hormones are involved. Hormones such as epinephrine, thyroxine. Neurohormones from the posterior pituitary gland such as oxytocin or antidiuretic hormone. Neurotransmitters such as epinephrine, norepinephrine, acetylcholine, dopamine, and the list goes on. When it comes to cellular reproduction, we have the cell cycle. And human cells, they don't last forever. They're subject to wear and tear. And most don't last for the lifetime of the individual. In most tissues, new cells are created as fast as the old cells die. And reproduction of gametes, sperm and egg cells, occur through a process called meiosis. The reproduction or division of other body cells, somatic cells, involve two sequential phases, mitosis or nuclear division, and cytokinesis and cytoplasmic division, which indicated in the diagram prophase, metaphase, anaphase and telophase. The phases of a cell cycle are a series of events that take place in the cell as it grows and divides. The cell spends most of its time in what is called interphase. And during this time, it grows, replicates its chromosomes, and prepares the cell for division. The cell then leaves interphase and goes into mitosis and completes its division. The resulting cells following this process is known as the daughter cells. Each enter their own interphase and begin a new round of the cell cycle. Replication, cellular reproduction. Now, cellular reproduction involves 5 phases. Prophase is the first phase of mitosis, and this is when duplicated genetic material is carried in the nucleus of the parent cell into 2 specific individual daughter cells. In metaphase, the process of cellular division, mitosis, chromosomes spread out in the cell nucleus and move together, aligning in the center of the dividing cell. In anaphase, this is 2 phases. Anaphase A, the chromosomes move to the poles, and in anaphase B, this is where the spindle poles move apart. The next phase that is gone into is telophase, and it's the last phase of mitosis, and it separates the duplicated genetic material carried in the nucleus of a parent cell into 2 individual identical daughter cells. And then the final step is cytokinesis of the cell cycle, in which the cell must faithfully separate the chromosomes and cytoplasm into 2 daughter cells. Now, although the complete cell cycle lasts 12 to 24 hours, about 1 hour is required for these 4 stages of mitosis and cytokinesis to occur. All types of cells undergo mitosis during formation of the embryo, but many adult cells such as nerve cells, lens cells of the eye, muscle cells lose their ability to replicate and divide. The cells of other tissues, however, particularly epithelial cells, cells of the intestine, lung, skin, divide continuously and rapidly, completing the entire cell cycle in less than 10 hours. Now, there are certain growth factors that are essential to cellular replication, and these are the cytokines. Growth factors are also called cytokines, and these are peptides that transmit signals within and between cells. And their major role is tissue growth and development. And having nutrients is not enough for a cell to proliferate. It has to also receive stimulatory chemical signals which are related to their growth factors. And these come from other cells or surrounding tissue called stroma. Examples of growth factors are platelet-derived growth factors, epidermal growth factor, insulin-like growth factor, and the list goes on. When we look at membrane transport, cell membrane transport, plasma membrane transport, we look at now the cell survival and growth, which is very dependent on the constant exchange of molecules within their internal and external environment. Cells continually import nutrients, fluids, chemical messages from the extracellular environment and expel metabolites, waste products, or products of metabolism out into the interstitial space. Now, when we look at membrane transport, we've got passive transport, which is diffusion. And this occurs across a lipid bilayer of plasma membrane for things such as molecules of oxygen and carbon dioxide. Then you've got active transport, and if there's activity, it requires energy. And these are pumps like the sodium-potassium pump, sodium-calcium pump, numerous types of pumps that are located within the cell membrane. Then you have membrane-mediated transport proteins for ions such as sodium, potassium, calcium, and magnesium, and the list goes on. Now, there are transporters, and transporters are ions that must fit exactly into a unique protein receptor. It's like a lock and a key. Then you've got channels. Channels have got certain electrical charges that open the gates. So, for example, there are sodium channels that can only be opened by a positive. There are channels that are related to ions that have a negative charge, like chloride. Body fluids are another component, things like electrolytes and solutes. Now, a body fluid or body fluids are composed of electrolytes, which are electrically charged particles that dissociate into constituent ions when placed in a solution. And then there are also non-electrolytes such as glucose, urea, creatinine, and uric acid. Electrolytes that exhibit polarity by orientating themselves towards a positive pole or a negative pole are examples. Ions with a positive charge are known as cations. Sodium is an example. Ions with a negative charge are known as anions. Chloride is an example. Electrolytes are measured typically in milliequivalents per litre or milligrams per deciliter. So, for example, your serum sodium has got a normal range of between 135 to 150 milliequivalents per litre. Your serum chloride has got a typical range between 96 and 106 milliequivalents per litre. Anions and cations vary in volume, quantity, size, depending on whether they are extracellular and intracellular. So, for example, you can see that extracellular sodium is 135 to 150 milliequivalents per litre. Intracellular, it's pretty small. Intracellular sodium is only 10 to 14 milliequivalents per litre. If we look at potassium, the extracellular potassium is pretty small, 3.5 to 5 milliequivalents per litre. Intracellular, it's incredibly high, 140 to 150 milliequivalents per litre. So these anions and cations vary depending on which compartment that they are located in. When we look at passive transport mechanisms, diffusion is probably the most common. And diffusion is the movement of a solute molecule from an area of greater solute concentration to an area of lesser solute concentration. And the difference in concentration is known as a concentration gradient. And it moves from high to low through a semi-permeable membrane. Another passive transport mechanism relates to filtration. Filtration is the movement of water and solutes through a semi-permeable membrane, and this is a result of now hydrostatic pressure. And hydrostatic pressure is a mechanical force pushing the water against cellular membranes. So for example, in the cardiovascular system, hydrostatic pressure is the blood pressure in the blood vessels when the heart contracts. And then when the blood eventually reaches the capillary bed, there's a hydrostatic pressure of approximately 25 to 30 millimeters of mercury, which is sufficient. It's a sufficient force to push water across the capillary membranes in the interstitial space. When we look at another passive transport mechanism, we can look at osmosis. Now, osmosis is the movement of water down a concentration gradient across a semi-permeable membrane from a region of higher water concentration to one of lower concentration. Now, for osmosis to occur, one, the membrane must be more permeable to water, not to solutes. Solutes can't go through the membrane. Water can. Two, the concentration of solutes in one side of the membrane must be greater than on the other side of the membrane, so that water moves easily from a low to a high concentration of solutes. Now, active transport mechanisms are different. Now, with active transport mechanisms, you can have endocytosis, where the cell creates a vesicle or a mouth. Endocytosis is when the plasma membrane enfolds substances from the outside of the cell, surrounds them, invaginates, and separates them from the plasma membrane, forming a vesicle that moves into the cell. And there are two types. You've got two types of endocytosis, and they're designated based on the size of the vesicle or the chamber that it forms. You've got pinocytosis, which again is the cell drinking. And this involves ingestion of fluids, bits of plasma membrane, and solute molecules through the formation of small vesicles. Then you've got bigger ones, phagocytosis, where the cell is eating. And this involves ingestion of large particles, such as bacteria, through the formation of large vesicles or mouths or vacuoles. Exocytosis is also with vesicle formation. In the eukaryotic cells, this is now the secretion of macromolecules, and this usually occurs by exocytosis. And the two main functions of exocytosis are replacement of portions of the plasma membrane that have been removed by endocytosis, and the release of molecules synthesized by the cells in the extracellular matrix. For example, could be waste products that are being released into the intracellular or extracellular matrix or compartment. Okay, that's the end of this session as an overview. Thanks. If you found this Dr. Manny Learn Shop to be of any value to you, please recommend it to your colleagues or friends. And if you want to be notified or be aware of other Dr. Manny Learn Shops, please subscribe. Take care and God bless.